So where we left off, we had looked at what happens in a monohybrid and dihybrid cross, and we went ahead and looked at this page briefly and showed that we could get a 9:3 to 3:1 phenotypic ratio from Mendel's P's. We went through a couple of other methods of getting there, so you didn't have to always do that big, gigantic dihybrid cross Punnett squaring. However, there's another possible outcome that could have come out of these. So if we come back and look at Mendel's cross that was done here, Mendel crossed true breeding double dominant P's with true breeding double recessive P's. If these alleles were inherited in such a way that they came down from the parent generation on the same piece of genetic material, or if somehow they were the gene responsible for these actually was one gene that caused multiple effects, then we could have had something far simpler come out of this. If these were under the rules of what's called dependent assortment, and whatever is causing the seeds to be yellow and round always tends to stick together, then we could have seen that we could have gotten a 3:1 ratio and only seen dominant or recessive traits come out in our phenotypes. Why didn't we see that? Well, Mendel found out that with these traits in particular, they are assorting independently, which means that the gametes are getting divvied up independent of one another. So the seed color and seed shape apparently were not linked genes, as we would call them. So when can genes be linked? Well, it would be when they come from the same region of the same chromosome. And if they're on the same piece of DNA as it would be here, and they're really close to one another, then they can be inherited together. And we say those genes are linked, and they tend to produce outcomes that are really close to this one on the left. They're not going to be exact always, but they'll end up closer to the Punnett square on the left than the one on the right. So this only happens when, in terms of independent assortment, things are truly independently assorted if their traits are on different chromosomes. So if the particular loci for these are on, say, chromosome number 1 and chromosome number 6, they're going to get independently assorted during what part of cell division? And if we are talking about traits that are on the same chromosome, we can have their loci being far enough apart to make them assort independently only if what event happens during prophase 1 of meiosis 1. Good job. So in dealing with your probability rules and your chi-square penny lab, you guys can probably answer these if you've already done the lab. If you flip a coin once and it lands heads, what's the probability of landing tails? Still 50%. If we cross two heterozygotes, what is the likelihood that the first offspring shows the dominant phenotype? It doesn't matter if it's first, last, middle, all of the odds are the same. It's going to be a 3/4 dominant phenotype if we cross two heterozygotes, just given by our Punnett square. We're going to have one double dominant, two heterozygous that show phenotypically dominant, and then one homozygous recessive. So we're going to see a 3:1 ratio there. And that's the same odds for the first offspring, the second offspring, the 100th offspring. It'll always be 3:1. And then same one here on this third question. If 4 offspring in a row express the dominant trait, what's the likelihood that the next offspring will express the recessive trait? It is the same rules. So we can show our pennies generally represent what's going on inside distribution of alleles, because there is a 50-50 chance of that allele, or if you think about us as diploid organisms, we're going to only pass on one particular chromosome for each homologous pair. so long as we've got this monohybrid cross under control, the rest of figuring out any bigger crosses is going to be easy. And I know all of you can do the monohybrid cross. So let's take a look at one that looks like it could be more complex. So we're doing in this case, we've got a trihybrid cross shown here at the bottom. And we're going to make it easy using 3 monohybrid crosses and then just use some fractions. So if you can take a look down here, we've got a trihybrid for pokey trait, which would be thorns, yellow trait, which is going to be yellow or green, and then the R trait, which is going to be round or wrinkled. So, and I'm curious here, what is the likelihood of a thornless green round offspring? And that would be it would be recessive in the case of the thorns, right? Because it would be not pokey. So we're looking for little p. It would be recessive in the case of the yellow or green traits. So we'd be looking for little y. And we'd be looking for a round offspring. That is a dominant trait. So we'd be looking for anything with a 1 dominant R allele. And we could make this into a big, huge Punnett square, but nobody wants to do that. That's terrible. Well, except for James Wilson, because he really enjoyed making a giant Punnett square. So on the rest of these, let's go ahead and break this down into 3 small Punnett squares. So do me a favor, if you can draw a Punnett square for just the p's, lay that out, and your Punnett square for just the p's is going to have big P, little p. I know I told you never to use p's because they all look the same. And what's the likelihood of us getting a thornless offspring out of here? That's right, 1/4, because it's just down here. That's the only place where we're going to get that one. So we don't need to worry about any big square. We've already got that handled. Let's take a look at the likelihood of us getting a green offspring. Again, this is the same thing with big Y's and little y's. Where are we going to get a green offspring? Again, right down here. That's the only place we're going to see it. And then finally, let's take a look at the likelihood of us getting a round offspring. So that one's a little bit more, not too much more complex, because round is dominant. And so we're going to be round here, round there, round there. We're only going to get this, 3/4 of the time. So now that we've got that accomplished, all we need to do to figure out the likelihood of all 3 of these things happening at the same time is to multiply the values together. So from the thornless, we had 1/4 times. From the green, we had 1/4 times. From the round, we had 3/4. That's all we need to do on these. So in this case, we end up with multiplication problem, and we just multiply straight across. We multiply fractions. So 1 times 1 times 3 is 3, and 4 times 4 is 16. 16 times 4 is going to be 64. So we end up with 3/64 of the offspring who are thornless, green, and round. And we could do this for all the other characteristics combinations that we could come up with, but we've really saved ourselves making a gigantic Punnett square and then crossing our eyes trying to figure out what's going on with the traits in here. Where do we find the thornless, green, round offspring? Because those can be kind of hard to find. I don't know how to get rid of the writing on my screen, so I'm going to pause here. So where did all the variations of different alleles originate? They originated with mutations, of course. So we are all different mutants of past alleles. And those mutations sometimes end up expressing or producing some sort of new protein, and those are dominant alleles. And sometimes those mutations end up being a loss of function, and those are recessive alleles. So coming back to the idea that genes don't interact with one another, dominant is just when it's producing some sort of a product or outcome. Recessive is just when there's not a product or an outcome. So if we look at a couple examples, Tay-Sachs disease, there is a dysfunctional enzyme that is supposed to, distracted because my wife was being mean and laughing at me.
Mutations, of course.
Well, I'm going to pause it and ask a question, a multiple choice question. Shh, I don't like you. You need to stop being so mean to me.
You need to stop being so funny. Mutations, of course.
Ugh.
Would you like to go?
Yes. Junior, scones are ready. Audrey, come get your scone. They're different from the other ones, though. I like it, though. It's good, though. Really good. It's not as crunchy as other ones, though. It's more doughy on the middle. That's okay. I still like it. Thank you, dear.
You're welcome. I'll try not to laugh at you, but you're going to have to make it harder for me to not laugh at you. Mutations, of course. And then I'm a teeth.
Really good.
Probably could be in the other for like 2 minutes. I'm just going to dry them out. But so now they're not jackhammering? Or that's only something you do at 7:30 in the morning?
Mm-hmm. I'm curious what they're doing.
Me too. Didn't have that much around the pool.
Well, and they drove that bobcat all the way out front. Now they have it back. I wonder if they carried something in with it, like a new pool pump or something.
I don't know. I don't know them long enough to ask. Son! What?
I did the thing with the thing in the middle.
Yeah.
You did that or you did it?
I did.
Yeah.
Okay.
Now you just have to peel off those middle stickers.
No, but she's already, Mom.
Good job.
Are you ready for Millwellens? The penguin guy. Or the pigeon guy. I'm putting it on the big TV. Go get your stuff to draw.
What is it?
I'm not on YouTube. It's being drawn. Go get your stuff to draw.
So in the context of dominant and recessive traits, Tay-Sachs is a good example to go through. So Tay-Sachs is a lysosomal storage disorder. We learned about it in our cells unit, and it can present as dominant, codominant, or recessive, depending on what traits you inherit and what level we look at. So Tay-Sachs at the organismal level, the recessive allele that is, if you have a recessive allele, it's not making the protein that it needs to make to be able to degrade lipids, and those lipids build up, and you end up having a recessive phenotype. Biochemically, however, we can have an incompletely dominant phenotype. So if you have a dominant allele and a recessive allele, the dominant allele is going to be transcribed and make proteins that have the job of degrading those lipids. And the recessive allele, instead of getting transcribed and making proteins, you're not going to get any. You only have half as many proteins doing their job as somebody who is homozygous dominant. However, it doesn't really matter, because as long as 50% of the product is there, it's enough. So we talk about that as being sufficient to still give you the dominant phenotype. So biochemically, though, we can say the enzyme activities are only incompletely dominant. We've got half as much of that particular enzyme being built. And then molecularly, we could say there is codominance happening here, because molecularly we are making two different versions of an enzyme. Those both end up getting built, just one of them is built and doesn't work. One of them is built and does do its job. In the context of dominant alleles and recessive alleles, so dominant and recessive alleles, dominant are not stronger, better, any faster, anything like that than recessive alleles. They're also not necessarily more common. Just because there is a dominant allele in a population does not mean it's going to take over. So dominance does not mean that evolution is going to happen, and it does not mean the recessive alleles are going to disappear. For instance, one of my friends has this allele. She has called polydactyly. She was born with 6 fingers, 6 toes on each one of her limbs, and one of her sons also picked up the trait. It's a dominant trait. She has one copy, and one of her sons has that one copy. They had to snip all the little nubbins off. It's not going to take over the population. It is not necessarily going to give somebody a selective advantage in sex or survival to have an extra little hot dog finger hanging off of the edge that doesn't really do anything. Okay? So dominant alleles are just dominant because there was a signal in that case to build an extra finger. And the presence of that signal caused an extra finger to get built. It did not mean that their gene was stronger or taking over the other genes better or anything like that. So that said, we can take a look at our next couple topics. Pleiotropy is when there are multiple outcomes from one particular gene. Cystic fibrosis is a fantastic example of this one, unfortunately. So in cystic fibrosis, there is I don't know if I can move my thing. So there is a protein that is built. It's in some cell membranes that are in mucus-lined membranes around our body. The ones that are in our chest, for example, in our lungs, have a mucus membrane that's supposed to be moist, and it helps for absorption of an exchange of gases, and it helps to also keep mucus moist and be able to move around and get out of our lungs as it gradually cleans us. If you have the bad genes for cystic fibrosis transmembrane regulator, or that gene, it ends up not just making you have problems with your lungs, but problems with your digestive tract and problems with your reproductive tracts, because all of those areas have mucus-lined membranes where stuff needs to move along them smoothly, and without that protein in place, you don't do what you're supposed to do. So sickle cell anemia is the next one we're going to look at. Sickle cell anemia is another example of pleiotropy. Just like cystic fibrosis, it is produced by a single nucleotide polymorphism. So there's one spot in the DNA that is messed up. It changes hemoglobin's shape, and after changing hemoglobin's shape, it produces a broad number of phenotypic outcomes. In the case of sickle cell anemia, it all relates back to the function of those red blood cells, and when they change shape and make them, instead of a nice donut shape, they get changed into a crescent shape or more of a sickle. Just like cystic fibrosis, there's one gene that is having multiple effects. In this case, there is one gene that is having multiple deleterious effects, and you can see on this graphic that both cystic fibrosis will eventually kill you due to digestive or lung issues in most cases. Sickle cell anemia will eventually find a way to get you because of all the blood and circulatory problems that it produces. So sometimes we have multiple alleles or multiple genes that interact and affect either single traits or multiple alleles that interact and affect a broad number of traits. So if we have multiple genes turning on or off a single trait, we call it epistasis. Epistasis literally means standing upon. So and we're going to look at a couple examples of this. Epistasis can really be explained well with things like coloration. So we've got coloration that can occur, and if the coloration is light or dark, we've got a couple of different alleles for that. However, there's a third enzyme that's responsible for making the pigment in the first place. And if the pigment is not made in the first place, it doesn't really matter if that pigment is destined to become brown or destined to become black or destined to become yellow, because there's just zero pigment made at all. So in our example of our Labradors, we get a very distinct ratio in terms of our phenotypes because we end up having, in this case of a dihybrid cross, we don't have a 9:3 to 3:1 ratio anymore. Because if you are a recessive for the allele that is responsible for making pigment in the first place, it doesn't really matter if you have brown or black pigment that's going to get made. You're just not making any pigment at all. So any of these that are missing the allele that's responsible for making pigment end up being the yellow labs, or we could call them white in terms of this particular pigment that's being looked at. And then the black and chocolate labs are the result of they're making the pigment. And then once it's down to that, what's going on? How are we doing this? Again, we could break this apart and do it as a part of two mini Punnett squares. If we were crossing a dog in terms of this E trait, how many of the offspring would be recessive? 1/4. Ah, well, we can see that 1/4 of their offspring are going to lack pigment altogether. 3/4 of them are going to have pigment. How many of them are going to have pigment and be a recessive for the brown instead of black? Well, it would be 3/4 times 1/4. 3/4 times 1/4 is, and you can see that on the screen. In terms of polygenics, sometimes polygenics are responsible for things that deal with numbers. For instance, height or skin color, where we have a whole bunch of genes and there is a gradual progression from one end of the spectrum to another. And this is from multiple genes, multiple alleles that are responsible for this. So in the case of skin color, we can see that there are at least 3 different markers that are responsible for this. And if there are only 3 different markers that are responsible for this, we have these 6 different combinations that we can get out of this big cross here. So So we can see that we've got multiple ways that we can inherit traits. Not everything is inherited, however. One other component is that we have environmental influence on our genes, and we have probably the best evidence for this in the case of identical twins. We all know some identical twins. Some of them are more identical than others, but by the time they develop into adults, there have been a number of environmental factors that have influenced them, from diet to mutations that have changed their own genomes, to other chemical agents that have potentially turned genes on or off. And through that, they end up developing differently. So we know that they started off the same, but their environment has shaped who they become and how they develop. So because of what we call polygenic characters, genes that traits that are that are the responsibility of multiple genes, we call them polygenic, and some of those polygenic ones are environmental. We end up getting a big multifactorial result in terms of the final phenotypic outcome. So human traits do follow Mendelian patterns of inheritance a lot of the time. I'm going to share a document with you guys to be able to look at a few of those examples of human traits. But humans are not ideal subjects. Why aren't they? And you guys have already looked at pedigrees in terms of our blood typing experiment. So pedigrees show usually something like this. So our males are square, females are circular. We usually shade in the trait that we are particularly interested in a pedigree. And then the genotypes may be filled in or deduced, or sometimes you can see on here they're left as a little bit of an option. We're not sure what the genotype is for some of these individuals. In a pattern where there is some skipping of generations, we can say for certain that there is a recessive sort of inheritance. And in patterns where the trait seems to follow through every single generation and never skips, that's when we can make the assumption that it's probably a dominant phenotype. We're not going to be able to breed humans hundreds of times to be able to really get good data on dominant recessive. So we're relying on just pedigree data for this. Not sure why I did that. Albinism is another great example of a Mendelian cross that you can do with humans. We've got people who come out of a family with albinism, but nobody has had it for multiple generations prior. It's got to be a recessive trait. And we can see in larger families of this, it does end up being about 1/4 of the time that child ends up with their lack of pigmentation. And this is another allele that is pleiotropic. We don't just have lack of pigment being made in the skin. There's also lack of pigment being made in certain parts of the eye, so they have vision problems, and lack of pigment, obviously, in the hair. So recessive alleles that cause diseases, if they're relatively rare, the odds of getting these are particularly low, unless we start doing inbreeding. And inbreeding, the primary problem with inbreeding is that there are a lot of recessive alleles out there, loss of function alleles, that are floating around in our population that aren't really common. But if you mate people that are really closely related, the odds of that coming to light are a lot higher. I think that has reached the end of a couple of our more questions. So, heterozygotes in sickle cell anemia. Sorry. So as most of our discussions in class have always ended with a discussion of evolution and the relatedness of things, we've got an interesting example of evolution going on with blood types right now. And we also have a couple of other interesting examples that we've mentioned as part of the note, so I wanted to make sure to hit on them. So cystic fibrosis, we've already seen, is a loss of function recessive phenotype. It is really common in Europeans or people of European descent. So it turns out that the people who have cystic fibrosis, historically, until recent modern medicine, have had a very short life expectancy. So this has been a death sentence for people. Recently, these people can live longer into their 40s and 50s with medicine, antibiotics, surgeries, and some pretty cool gene therapy that's being done right now, where they can actually modify the badly made proteins by editing some of the genes within these adults. So there's some cool biotech involved with this. However, the reason it's super common among Europeans, a bit of an interesting mystery. Turns out this is an example of what's called heterozygote advantage. In some cases of sicknesses that wiped through Europe or went through Europe, people who were heterozygotes and only had 50% of their chloride channels functioning to be able to moisten up the mucous membranes in their body could not drown in pneumonia, flood their lungs with water, did not have significant quantities of things like diarrhea that the homozygous dominant people did. So these people had an advantage. Obviously, the recessive, homozygous recessives were going to die, but homozygous dominants also died, and then these became instead of a relatively insignificant, rare part of the population compared to the rest, well, once the rest is killed off more largely, now these are a much bigger piece of the pie. Sickle cell anemia is another one where it has some evolutionary impact on the human record. This one is really common in just African Americans. So how do you get a group from, and this is not African Americans only, it's people of all African descent. Why do you get people who have this coming from just one area? And why is it so common? Well, could have been something that just arose in early African people, or it could be something that was an advantage to have at some point. Again, sickle cell anemia, death sentence, especially before modern medicine and people recognized what it was. So you can see the symptoms on here are not pleasant. However, people who are heterozygotes, they have minimal symptoms. And why is this? Well, heterozygotes in this case are less susceptible to getting malaria. Something about their change in shape of the hemoglobin protein allows malaria to be eliminated from the bloodstream. And malaria is carried by the Aedes aegypti mosquito. So mosquitoes carry this guy and distribute it among people. So why is this gene so common in Africans? And then some dominantly inherited disorders. Now these are super rare. For dominant alleles that cause a lethal disease, why would that be? And why is it that they not only are rare and they do not tend to get inherited? Why are they usually a new mutation that comes up? So we've got dwarfism is relatively rare, and it's caused by a dominant allele. Huntington's is another example. So dwarfism doesn't kill you. Huntington's does, but Huntington's doesn't hit with any sort of effects until you're 35 to 45 years of age. Why is the timing of that particular one really sad and disappointing?